Alrighty, students, welcome back to Chapter 4, which focuses on kind of introducing you guys to the cell. We've covered the introduction to the cell, compared prokaryotes and eukaryotes. We've discussed the nucleus and ribosomes, and then also the endomembrane system. So this lecture is going to focus on our energy-converting organelles, and then also the cytoskeleton and cell surfaces. So let's start with the energy-converting organelles first. There are two of them. The first, which is in all of the eukaryotic cells, are what we call mitochondria, mitochondrion singular. And what essentially mitochondria do is something called cellular respiration. It's essentially, it's actually aerobic cellular respiration, so it requires oxygen. We take nutrients like sugars and carbohydrates and proteins and things like that, and with the aid of oxygen, aerobic, we take those nutrients and we turn them into energy, usually in the form of a molecule called ATP. Adenosine triphosphate. So mitochondria have kind of a very specific shape to them. They have kind of two internal compartments. So they have an outer membrane and then an inner membrane. Between the outer membrane and the inner membrane is this sort of narrow intermembrane space. And then inside the inner membrane is really where we find what we call the mitochondrial matrix. And that is where the inner membrane is sort of arranged in these kind of shelf-like structures called cristae, and this is where that cellular respiration takes place. So there's enzymes that help that happen. You'll also find the mitochondrial DNA and the mitochondrial ribosomes, because mitochondria have their own DNA and their own ribosomes. The other energy-converting organelle is found only in the plants and the algae, so not in animal cells. And it is known as the chloroplast. And its job is photosynthesis, which is essentially the conversion of the energy from the sun into the chemical energy in the form of sugar molecules. So plants and algae essentially get to make their own food. They don't have to ingest food like we do. They also have a very kind of specific structure to them. You can see they've got two membranes as well. And then essentially they have these little stacks of granum and thylakoids, almost like little stacks of poker chips inside the two membranes of the chloroplast. At least that's what they make me look like. That's what they look like to me, little poker chips. Now, I mentioned the fact that mitochondria have their own DNA. Well, so do the chloroplasts. And the reason the mitochondria and the chloroplasts have their own DNA, unlike all of the other organelles inside the cell, is that they were once free-living organisms. It's called Endosymbionic Theory. The mitochondria and the chloroplasts were, at one point, these very small prokaryotic cells that essentially began living inside a larger cell. And the larger cell found the kind of energy using and kind of prokaryotic, what becomes a mitochondria, quite useful. As the atmosphere was getting more oxygen in it, this eukaryotic cell essentially got energy from the mitochondria, and that was quite useful for the larger cell, but the mitochondria, the little oxygen using protist, got some food essentially from the bigger cell. And so it was kind of a mutually beneficial arrangement. And eventually the mitochondria lost the ability to live on their own and can now only exist inside the eukaryotic cells. So all cells have mitochondria, which means that this endosymbiosis happened not once, but actually twice for some organisms. So animal cells, protists, things like that, no chloroplasts, only the algae and plants. But in some eukaryotic cells in the ancestry, they got another endosymbiont in them, one that did some photosynthesis, and eventually that kind of photosynthetic prokaryote again lost the ability to live outside itself and became a chloroplast. And so all eukaryotic organisms will have mitochondria, but not all eukaryotic organisms will have chloroplasts. That means the mitochondria developed first in our evolutionary history, and the chloroplast developed second. So the chloroplast is a newer adaptation than the mitochondria. All right. The next thing we're going to talk about is actually the cytoskeleton. So if you think of our bones inside of our body that give us all that kind of structural support, well, cells sort of have the same thing. It's not quite as hard as ours is. It's more of a soft skeleton, but they do have a cytoskeleton. And then there are also some modifications that happen to the surfaces of our cells, and we'll talk about those as well. So the cytoskeleton has essentially 3 different sets of proteins in it: microfilaments, intermediate filaments, and microtubules. So the microfilaments are the smallest, the microtubules are the biggest, and then intermediate, of course, falls in between. They help cells maintain shape. They anchor and move organelles around the cell. Sometimes they will give a little what we call amoeboid movement to cells, which is this sort of like squeezy, kind of gelatinous, kind of slow, kind of uncontrolled movement. And then the cytoskeleton can also sometimes function in contraction of our muscles. So you can see here are our 3 different types of cytoskeletal elements. Here is the microtubule. It is made up of the protein tubulin. It is the kind of the biggest of the bunch at 25 nanometers. The microfilaments are made up of a protein called actin. It is the smallest at 7 nanometers. This is what a good chunk of our skeletal muscles are made from. You can see these tubulins making a sort of kind of web of skeleton there. And then intermediate between the microfilaments and the microtubules are these intermediate filaments. They are made up of these fibrous proteins that essentially get coiled together kind of like rope, and so they act like guy wires inside the cell. So let's think about a kind of big picture here, right? So it wasn't really until the 1940s that scientists actually were able to identify 2 proteins that make up our skeletal muscles. So the muscles that move us, right, the kind of the meat of our body, it's 2 tiny little proteins called actin and myosin. And again, we really didn't know about these until the 40s. And it wasn't until, mid-50s or so that, using some more advanced microscopes, scientists were actually able to determine how the actin and the myosin interact during muscle contraction. And it's like this kind of super kind of interesting process where the myosin sort of grabs onto the actin and kind of ratchets it into the middle of the cell. If you've like used like a ratcheting screwdriver, it sort of works like that. And then after we discovered the myosin and the actin in the muscle cells, scientists were able to discover actin filaments in all types of cells, not just our muscle cells. They then kind of tagged these actin molecules with some fluorescence and then injected it into other living cells, and were able to really get some just amazing visualization of the behavior and interactions between the different cytoskeletal elements inside our cells. And so here we're using a little biochemistry, a little microscopy to really study anatomy, which is pretty cool. So let's talk about some surface structures. 2 of them are sort of locomotor appendages, so used for movement. These are cilia and flagella, flagellum singular. Cilium singular. So these are composed of microtubules. The flagella is longer. It's sort of a whip-like motion. the cilia kind of work together. If you've ever watched a crew team, a rowing team, when they got to row together, the cilia sort of work like that. But they are a 9-tube arrangement of microtubules. So you can see here's the little central microtubules, and then the outer microtubules in these little kind of couplets. You can also see they've got little proteins that connect it all together, and then what are called dyneins, which are essentially these motor proteins. And it's really those motor proteins that allow the cilia and flagella to have that locomotion and that movement to them. And you can see how tiny they are. This is a transmission electron microscope image, and we're nearly 300,000 times the size of it, the actual object. So we're talking about really, really, really microscopic microscopic structures here, beyond microscopic structures. So let's again kind of look at a bigger picture issue, right? There is a fairly rare disease, but does occur. It's called primary cilia dyskinesia, PCD. Essentially, the cilia are immotile, meaning they don't move like they normally should. And then the flagella themselves also don't have any motor proteins. This disease, PCD, has people who have PCD very often have respiratory tract infections and sperm that are non-motile, that don't move. And that seems kind of really unrelated, right? Like respiratory tract infections and infertility. Like how do those kind of, like how does one disease result in both of those symptoms? I want you to kind of think about that for a second. And think about what the name of the disease is. Right? Dyskinesia. Right? So our respiratory tract is lined with cilia. It's a protective mechanism. Our respiratory tract makes lots of mucus, and the cilia help bring up that mucus, which might contain bacteria or viruses like SARS-CoV-2 from infecting us. But when the cilia are not motile, the cilia can't move, then it makes that respiratory tract much more prone to infections. Now sperm don't have cilia, but they have flagella. And so if you have flagella with no motor proteins, then the sperm are not going to be able to move, they're not going to be able to swim, and that person will have some fertility issues. So this is a disease, PCD, which can result in very disparate systems or symptoms, but really with the same cause, which is the kind of no motor function for these two cellular extensions. Now, thus far, we've been talking about inside the cell, all of the organelles intracellularly within the cell. But animal cells, or some animal cells, a lot of animal cells actually, secrete stuff extracellularly outside the cell. It's called the extracellular matrix. It's sort of this gelatinous kind of fibrous kind of glue that binds tissues together, that helps support our plasma membranes, and that also communicates kind of back and forth with the cytoskeleton inside the skull. And it actually may attach to the cell using what are called glycoproteins, which is just a fancy word for sugar proteins, that help bind them to proteins in the plasma membrane of the cell. It's called an integrin. And so it helps kind of keep the cells together. So here you can see, here is our cell. Here you can see some microfilaments of the cytoskeleton. But here we have all this kind of extracellular stuff. We've got these big, long glycoproteins that are going to help connect to each other and to these integrin proteins. And then also these big kind of collagen fibers, and there's elastic fibers in here, and just all this kind of extracellular fluidy gelatinous stuff. There are generally 3 types of junctions that we find in animal cells: tight junctions, anchoring junctions, and gap junctions. So tight junctions and gap junctions are sort of at opposite ends of the spectrum. So tight junctions are sort of preventing fluid or other solutes from kind of moving across cells. So like you don't want to think about like zip blocking your bag real tight, right? So that nothing gets out of the cell or into the Ziploc bag. That's sort of like a tight junction. So it's sort of like a zipper, or like a Ziploc zipper. The gap junctions are sort of the opposite end of that. They actually want stuff, and they allow stuff to move pretty easily from one cell to the next. So it's more about kind of not preventing flow, but allowing flow. And then the anchoring junctions are like a rivet. They're like the stuff that you have on little kids' shoes. Velcro. Couldn't think of that there for a second, right? You know how Velcro kind of sticks to each other and it makes the Velcro sticks so that you can't pull it apart? Well, that's what these anchoring junctions help do. They help keep our cells really anchored and connected to each other so that they would not split. So here's another little kind of comprehension question. If you had a muscle tear, right? Maybe you tore your muscle. That would involve the kind of breakage, the rupture of which type of these junctions do you think? Would that be the tight junction, the gap junction, or the anchoring junction? A muscle tear. I'll let you think about it. It would likely be an anchoring junction because, again, the anchoring junctions are going to attach cells to each other into these really, really strong tissues that help the cells stay together. So the cell junctions here and the extracellular matrix are structures that we find in animal cells. So let's talk about some stuff that we find generally only in plant cells. One of these things is a cell wall, which is a rigid structure, much more rigid than the plasma membrane. So the cell wall provides sort of additional support, particularly, right, because we're talking about we have all these plants that have to stay upright on land. And if you've ever looked at the stem of a rose, right? It's pretty floppy. We need a little bit of extra support, and so we get these cell walls made of cellulose. Plant cells also have plasmodesmata, which are kind of specialized junctions found only in plant cells. These allow plant cells to share water, nutrients, kind of talk to each other, so on and so forth. So the plasmodesmata are most similar to which type of animal junction do you think? The anchoring junction, the tight junction, or the gap junction? Remember, here's your 3 animal cell junctions. If you said the plasmodesmata were similar to the gap junctions, you would be correct. So here you can see, here is our cell wall made of cellulose. And then you can see here's the little plasmodesmata, so stuff can pretty easily move from one cell to the other. It's just like our gap junctions. So this is table 4.22. It's an excellent table to review because it reviews all of the kind of different functional groups and the organelles inside those functional groups. Gives you a little kind of blurb about what they do, and then also a little bit nice little kind of diagram there about what they look like. So this is an excellent table to review. And then at the end of the PowerPoint file, you will find sort of the study guide, actually. If you think of it sort of like a study guide, if you can answer these questions, right? Describe the importance of microscopes, the parts of the cell theory, compare prokaryotic and eukaryotic cells, how cell size gets limited, structure and functions of cell membranes, compartmentalization in eukaryotes, structures of plant and animal cells, the different functions of chloroplasts in the mitochondria, endosymbiotic theory, the different parts of, compare and contrast the different parts of the cytoskeleton, relate the structure of the cilia and flagella to their functions, talk about the extracellular matrix, compare and contrast the three types of animal junctions, or the cells, the three types of junctions between animal cells, and then also relate plant cell walls and the plasmodesmata to their functions, and just overall describe the different categories of organelles and how they function inside the cell, then you are A-OK with Chapter 4.